presentation is about how visual tracking can be used to reveal foraging distributions around breeding colonies for 4 species of tern, and this uses a habitat modelling approach. So 4 species of tern are the subject of this presentation, and these are all listed as rare or vulnerable under the EU Birds Directive. So they already have colonies which are protected as part of a network of terrestrial special protection areas, or SPAs, but there are no marine SPAs which would protect their foraging areas at the moment. So the aim of this work was to identify the important marine areas for these 4 species of breeding terns in the UK. So first of all, we surveyed a sample of colonies, and then we analysed this information to characterise the marine foraging habitat, and then we could map the foraging distributions around colonies, including colonies for which we hadn't collected any data. We used a method called visual tracking, where we follow birds in a boat, and the GPS track from the boat acts as a proxy for the birds track. And because we have an observer on the boat, we're able to extract foraging locations from these tracks. So the map on the bottom left shows some example tracks from a colony in Northern Ireland, and these tracks are for Arctic terns, and they were collected over 3 years. So if you look at the map on the top right-hand corner, this is a simplified example showing how we treated this data for analysis. So in this example, the white line represents one track, and the blue dots along this line represent the locations where that bird was observed to forage. So these act as presences for the analysis. And the red dots, they're examples of locations where that bird did not go during that particular foraging trip, and these act as absences for the analysis. The analysis uses a logistic generalised linear model with the presences and absences and a range of environmental covariates. So that can be things like water depth, sea surface temperature, or distance to colony. And these are covariates which might represent either prey distributions or prey availability to the birds. And then the analysis calculates the probability of a tern foraging at a particular location. And these are within the species-specific known foraging ranges. So the map on the bottom right shows a simplified example of the kind of output that we might get. And we can turn these probabilities into relative foraging densities. And there's an example output for Arctic terns at Coquet Island again. So we undertake this analysis in 2 phases. In phase 1, we develop species and colony specific models where we have sufficient data to do so. So that will give you a nice detailed prediction of where the birds at that colony are going to forage. In phase 2, we develop generic models using information from the surveyed sites, and we can use these to make predictions of foraging distributions at unsurveyed sites. So as an example, we might have data for a particular species of tern from colonies A, B and C, and we can put that data together to produce a generic model which we could use to predict foraging distributions around colony D, for example. And we want to assess how well the phase 2 models in particular will be performing. And so we can use cross-validation to do this as part of our model development process. So this would, if we go back to the example of having data from colonies A and B and C, the cross-validation process would first of all build a model using only the data from colonies A and B, and it would use this to make predictions to colony C, and then we can compare those predictions to the actual data that we do have for colony C to see how well that model is performing. And cross-validation would do this for all combinations of colonies A and B and C. And when we're happy that our model is performing well, we then pool the data across colonies A and B and C to produce our final generic model for that species and use that to make predictions around colony D and colony E, for example. Across phases 1 and 2, we produced 45 foraging distribution outputs across the 4 species and 32 colonies. You can find more information on this analysis in JNCC Report 500, which you can download from the JNCC website, or by asking me questions. Thank you for your time.